The story of genetics is critical to understanding adaptation and evolution. Combined with time, this has led to the vast diversity we see in the natural world today. Among eukaryotes, the raw materials for evolution are provided by genetic mutations. Diversity is further generated through recombination and sexual reproduction. These are usually gradual processes, and their impacts are felt over long periods of time. But there are examples of radical changes occurring in a single generation. Not by changing the genes, but by changing how many of each gene are contained in a species. That is to say, by changing their ploidy. Defined formally, ploidy is the number of complete sets of homologous chromosomes in a cell. For example, we know human beings are diploid, having two sets of 23 chromosomes in each somatic cell. Our gametes are haploid, having only one chromosomal set each. There is great diversity surrounding ploidy in eukaryotes, with some species that are haploid, some that are diploid, and others that are polyploid, meaning they have three or more chromosomal sets. Although diploids are the most common, researchers think that many have polyploid ancestry. But ploidy is more than just counting a number of chromosome sets. Organisms can have different properties due to their ploidy. In particular, polyploidy can confer higher genetic fitness, genetic redundancy, and the ability to reproduce asexually. And while the development of polyploidy tends to be lethal in animals, other organisms, like plants, are more resilient. Today, the world population is estimated at 7.6 billion people. Slated to increase by 30% to 9 billion by 2050, food shortages are expected due to limited land, water, and energy. Impending climate change also threatens our food supply, with rising global temperatures leading to unpredictable weather patterns. But scientists are working to develop the next generation of crops to increase yields and adapt to changing conditions. The qualities that polyploidy confers to plants might be able to help us solve some of these food security issues. This is because polyploid plants can have a higher genetic fitness and greater resistance to drought, pests, and other stressful environments. Many of the foods we eat already come from polyploid plants. For example, peanuts are tetraploid, wheat is hexaploid, and strawberries are octaploid. Another polyploid plant is the coastal redwood, which grows over 100 meters tall and is extremely disease and pest resistant. Studying how polyploidy contributes to these qualities can help us develop more efficient disease-resistant crops, increasing food yields for a growing population. Beyond helping us develop new crops, understanding ploidy may also help us treat cancer. 1 in 3 Canadians will be diagnosed with cancer in their lifetime. With 200,000 new patients each year, cancer is the leading cause of death in Canada. Although each case of cancer is different, the uncontrolled cell division characteristic of cancer results in both diploid and polyploid cancer cells. Polyploid cancer cells are more likely to survive targeted therapy compared to the diploid cancer cells. The things that make polyploidy desirable in plants, that is, the genetic redundancy and ability to diversify, makes polyploid cancers all the more dangerous. Because of this, the cancer cells can grow unchecked despite treatment. However, polyploid cancers have special metabolic demands that make them sensitive to certain treatments. Scientists have identified some of these and are working on therapeutics to disrupt the specific metabolic pathways. So ploidy is more than just counting sets of chromosomes. Beyond ploidy's role in evolution, understanding how ploidy can impact survival and adaptation will be integral to surviving a changing planet and treating a major class of diseases.